Hello, everybody. Good morning or good afternoon or good evening, wherever you are. And welcome to this latest collaborative webinar between the Whitehead Institute, Memorial Sloan Kettering Cancer Centre and the Francis Crick Institute. I'll just leave that splash screen there for a few seconds. Those of you who've been to any of these webinars before will know that there's a couple of ways of contacting us. You can use the chat function, which is really useful if you want to say hello, or if there's any issues with our sound or anything like that. But if you have any questions, the Q&A button is the best place to put those. And what we will do is we'll attempt to answer all of your questions during the course of the next 90 minutes. We may not answer them immediately, and we may group some of them together as we go along, but hopefully we'll get to all of them at some point. So what are we going to talk about today? As you can see from the title there, we're going to go slightly differently to what we've done before. We don't have a machine in front of us today. We are going to do some live flow cytometric data analysis. And when I say we, that's me. So my name's Derek Davis. I am the STP, or Science Technology Platform Training Lead at the Francis Crick Institute in London, in the UK. And for many years, I have run a core flow cytometry facility, but now I look after education and training, particularly externally facing. And I'm joined here today by Kathy.
Thank you, Derek. So yes, my name is Kathy Daniels, and I'm the flow cytometry core facility here at the Whitehead Institute for Biomedical Research in Cambridge, Massachusetts, in the United States. I've been here for about 2 months now, so I'm brief in my tenure here. But I've been working in a flow cytometry core facility either at Sloan Kettering or before that at Regeneron for the past 8 years now. And I did get the privilege of working with Rui Gardner at Memorial Sloan Kettering Cancer Center. And unfortunately, he's not able to make it today, and he wishes he could be here, but we'll do our best to do him justice in his absence. So thank you.
He should have left us a cardboard cutout or something, isn't he, that we could at least put in the corner.
That'll be a special request for next time.
Okay, so as I said, we're going to talk about data analysis today. And in our previous webinars, we've covered a lot of the practical aspects of flow cytometry. So we've looked at QC, we've looked at setting voltages, we've looked at compensation. We've also looked at full spectrum analysis and cell sorting. And what we do in the core facility, a lot of our time is spent helping people think about their experiment. So designing the experiment, thinking about sample preparation, thinking about staining protocols, ensuring that they bring an optimized sample to the flow cytometer. We spend a lot of time also training people to run their data in an optimized way. And then quite often we let them go off with their data. Now, this is something I'd rather not do, but if you've got a multi-user facility, sometimes it's almost inevitable. But data analysis in flow cytometry is extremely important. Now, you're asking a biological question and you want to get an answer by flow cytometry. Your data file may contain that answer within it, but you have to dig for gold, if you like. Sometimes you have to do some panning to find the right answer within there. And what I found over the years is that users will go off, they'll go back to their lab who may have somebody who's experienced in flow analysis, and they'll teach them what to do. But that person was taught by another postdoc who was taught by a different postdoc who may have been taught by a flow cytometrist. But that chain is getting so long, it's a bit like Chinese whispers. And people will come to me with a question and we'll sit down and I'll say, right, let's open up this plot. And they'll say, that isn't the way I've been shown to do it. That's a different plot. You haven't put this against that. So what we want to try to get people to understand is the principles of data analysis. Now, what we're going to talk about today is actually going to be relatively simple, I think, because I've been doing this for a large number of years. But we're not going to be going and doing high parameter t-SNE analysis or anything like that. But the principles that we are going to look at today will hopefully help us in all types of data analysis. So what are we going to do? Okay, so we're going to go live in a minute, and today we're going to use FlowJo for our analysis. I might say this several times, but this is not going to be a FlowJo tutorial, and FlowJo isn't the only analysis software out there. In fact, in our next session, we will probably be using FCS Express to do this, and it doesn't matter which cytometric analysis program you're going to use. 95% plus of the time, they will do exactly the same thing, and certainly everything we're going to look at today, they will be capable of. So we're going to start fairly simple by looking at the basics of a flow cytometry file. We'll look at the types of plot that we can use in our analysis. We'll look at regions and gates and how we set them and why we might set them in particular places, and also how we display the data. So that takes into account things like data scaling. And hopefully we'll have enough time to do a couple of very simple examples. So I'm going to cover cell viability, and then Kathy is going to actually take some of the data that we generated in one of the previous courses, or at least some data that's very similar, and look at how we assess the correct voltage to set on the systems where we can set voltages. And as we said, questions are certainly encouraged all along the way. So Kathy will be monitoring those while I'm speaking, and vice versa. So let me stop the share at that point. Let's actually get on with things. So I'm running this off a laptop with a relatively small screen. So forgive me if I take a few seconds to find out what I'm doing. Okay, so I'm going to share my screen, but this time I'm going to share the whole of my screen. One thing I found with these sort of demos is that, can you see my screen, first of all? That's one thing. Kathy, can you just confirm that you can see that? Right, okay, good.
We can see the screen.
Good, nothing on there yet, not much on there. So I say that what these courses make me do is tidy up my desktop, because otherwise this would be a complete mess. So you can obviously see that everything's been piled in a folder called desktop over there. So as I said, what we're going to do today is we're going to use FlowJo, and you can probably see that I'm using a Mac here, and FlowJo will run on both PC operating systems and the Mac OS as well. Or it's certainly been able to do that since the inception of version 10 of FlowJo, because it's a Java-based program. There are some slight differences still between the PC and the Mac version. And here we are, then here is the FlowJo icon. And I'm going to be using version 10.7.1. This is almost the most up-to-date version. I believe there's a 10.7.2, but I always like to lag one version behind, because I know it works and I know it does what I want this to do. So if I click on that. So the versions of FlowJo can come in several flavors. You can have a dongle protected version, you can have a hardware specific version, or you can have, like we have at the Crick, you can have a site license for FlowJo. So that means for me to get this to work, I have to be connected to our VPN, but don't worry too much about that. So again, one thing I'll state again here, this is not a FlowJo tutorial. If you want to learn all the bells and whistles of FlowJo, please go to their website. There's a lot of great material there, and they're quite happy to reach out to you and show you what it does. But of necessity, obviously, I will be using FlowJo and parts of it, and we'll try and point out some of the important parts of that. But what I'm going to show you is common to all of the analysis software. So I have a floating window here. I'll try not to move that too quickly. I know some of you might be looking at this on a tablet, which means you'll get motion sick if I move that too quickly. But as well as the little buttons at the top here, which we'll come to in a little while, I have two areas here. The upper area where I can add as many samples or as many experiments as I like, and the workspace can hold a single experiment, or it can hold multiple experiments. Or we're going to actually keep it very simple here today. I can, as it says here, drag my samples into this area of the workspace, and that's where they'll end up. So I'm not going to do anything particularly complicated today. You can see I've got 4 files. Actually, I probably won't even use 4 for my part of the demo. But what I wanted to start with was thinking about how we can understand the structure of a flow cytometry data file and how we can start thinking about which types of plot that we can show to help us show our audience or our boss or our readers what we've done with our analysis. So I'm actually going to start with this file here, which says 8 peaks. So we talked about in one of our early webinars about how we always need to quality control our flow cytometers. And there are multiple ways of doing that. Most of the time we will use multi-peak beads because that helps us assess both the alignment of the lasers and the sensitivity of our detectors. But I'm using this as an example because you don't have to worry about any biology here at all. So I'm just going to drag that into here. You'll notice here that this doesn't say 8 peaks. I'm going to just add the file name in here. Don't worry about what I'm doing here, but this tells me, this gives me the actual name of the file because this is a Diva-based file. It wasn't exported correctly and it's been given the Diva name there. So I've got one file in here and you'll notice it says .fcs. Okay, so FCS stands for Flow Cytometry Standard. We may have mentioned this in passing before. And the great thing about flow cytometry data files is that every flow cytometer, no matter who makes it, will have the capability of exporting your data in flow cytometry standard format. And we're currently on version 3.2 of this. So this is something that came about through ISAC, the International Society for the Advancement of Cytometry. And this is great because it means that there's a standard format of these files. So companies like FlowJo, FCS Express, other programs like Kaluza, FlowLogic, Venturi 1, and so on, can all handle these data files no matter which flow cytometer they've come from. And one of the things about these FCS files is that they come in 2 portions. So we have a header file, which I'll come back to in a second, which holds information about the cytometer, and the data portion of the file. Now, the data in our flow cytometry standard files is really a spreadsheet. So as cell 1 goes through the laser beam, I get information about forward scatter, side scatter, and any of the different fluorochromes that I've measured. Then cell 2 goes through, I get the same information, cell 3, cell 4, and so on. So I end up with hundreds of thousands or millions of rows and multiple columns. So I could take that data and analyze it in Excel or Prism, or I could manipulate it in R. But the advantage for us as end users of having a program like FlowJo is that it will handle all that for me. And also it gives me a chance to explore that data. Now, I'm sure Kathy and Rui would say the same here, that if we are troubleshooting your experiment, what I don't want is a text description of what's gone wrong, or even a PowerPoint or a PDF, although they're a bit more useful. What I want is a data file, because if I've got a data file, I can go in there and I can find out something about it. So if I click on that data file on the Mac, go to Control, I can go to this Inspect tab here. And what that brings up is all the information that's contained in the header portion of my data file. So if we look on the left here, you can see the date that this file was run, the cytometer that was run on. So it's on a sorter, an ARIA3. All of these little things that begin with a $sign are keywords that are in the flow cytometry data standard. There is some leeway within there for different companies to put their proprietary information. So because this is a BD machine, it's got information about the CS&T, remember the cytometer setup and tracking program that allows us to QC the machine. But I can look at bits of information here. So I can look, for example, at the time that we started, not only the date, but actually the time that we, in this case, finished, where it started, it's up here somewhere, beginning time. So it took somebody 49 seconds to acquire the data here. There's important information about the laser delay here. This is a spatially separated system, so it's useful to know how many microseconds the electronics was working on to marry up our signals. So again, I can look at that and I can see whether that looks sensible or not. There's a lot of information that even I don't bother looking at. It also tells me something like the threshold that was used. Remember, the threshold is that hurdle that data has to jump over before we measure it. Now, this is a data file, a QC data file, so there was no compensation set, but if there were any compensation, that would appear here in the compensation matrix. One of the big plus points, I think, of modern day flow cytometers is that our data is all stored uncompensated, and any compensation you've applied at the cytometer is stored as a matrix. So what FlowJo would do, and what any of the other programs would do, it would take the data, it would take the matrix, and it would reapply that matrix in analysis. And that's great because it means that we have the ability to change that if we got it wrong. And compensation in offline software is something that we will look at in a future webinar. Also, very importantly for me, it contains information about the parameters that we measured. So all of the parameters here, so this is an 18 color cell sorter, so all of the channels are open. But the thing I'd be looking at is this, the voltage that's been applied to each of those channels. So I can very quickly scroll through there and I can see that nothing looks out of the ordinary. There's nothing that's a very low voltage or a very high voltage. So I'd be reasonably happy with that to start with. I'll come back to this a little bit later as well. So if I want to see my data, if I do here, FlowJo, I'll just double click on there and I get a plot, and it defaults to forward scatter and side scatter. And I can change the axes. So obviously this is a bivariate display. I can change the axes simply by clicking on here. So if I want to change that to the FITC channel, I click on there. Remember, this is an 8-peak bead set, so I can see my 8 peaks. So it's very easy to change that. It's defaulted to this what I'd call a polychromatic dot plot or a pseudo color plot. And many of you are familiar with these from either running your own samples or seeing these in the literature, where it's a sort of a combination of a contour plot and a dot plot, I suppose. So areas of high density are colored, so colored in red. I can see high density here will be in red. So although I've got two populations here, I know that actually in this population, there aren't very many events. And because these are beads, they're all small, round, uniform particles, I'm pretty sure that these are my singlet beads. These are probably doublets of bead. But this isn't the only type of bivariate plot we can show. So here in FlowJo, I can alter these. So I'm a big fan of these pseudo color ones. The other one I quite often use is a contour plot. So a contour plot is just like a map. It joins up areas of equal density, and we have the ability to change the sort of distance between the contours here. One of the slight disadvantages of a contour plot is it doesn't show us any of the data or the information that's below the first contour level. Well, actually, we can do that because I can click this little show outliers here, and that shows me all of those sort of individual events that are below the first contour. There are some other types of plot we can use. Occasionally, you might use a dot plot. Now, this is something that going back 30 or plus years, we used a lot of because it was the only type of plot we had. And because we only ever acquired 10,000 events, that's all we could fit on our floppy disk, that worked reasonably well. And if I've got a small number of events, dot plots can work. However, if I've got a lot of events, there are millions of them, this becomes very black very quickly and we lose any granularity. Kathy, you've appeared.
I have appeared. I think this is a good time as you're showing these different tools in FlowJo to bring up a question that one of our attendees has asked. So Krivono has asked, how common or uncommon is it to use BD-Fax Diva for analysis as well? So we're focusing right today on FlowJo for analysis, but
yes, I probably should have mentioned that at the beginning. Is you can do your analysis in the machine, on the machine that you've acquired your data on. Most of the acquisition programs have a degree of analysis in there, but very often they don't have the sort of bells and whistles that the offline programs will have. Diva, for example, you can't really overlay samples very easily. So normally we would try and take that data off and analyze it in an offline program. It's more convenient as well. And often you're paying for access to the cytometer. You don't necessarily spend a couple of hours doing your analysis there.
Agreed completely.
Right. Yeah, so dot plot. So there are a couple of other types of plot in FlowJo. We have this thing called a density plot and another one called a zebra plot. Both of those to me look like somebody spilt their coffee over there. But one thing I would always say is that you are in control of the way that you show your data. If this shows it better than another type of plot, hey, that's what you use. One word of caution, though, in that you could use any of these plots in a talk like this or a lab meeting. But if you want to publish the data, there are some journals, particularly the Nature Publishing Group, and hey, that's where everyone wants to publish, right, that will only accept the contour plot with outliers or the pseudo color plots, which is why I tend to use those almost exclusively. It just makes it easier when it's time to publish that data. Of course, we don't always want to show our data as a bivariate plot. Sometimes we want to show it as a histogram. So in FlowJo, I could scroll all the way down here to where it says histogram. The other thing I can do is I can type in histogram and it will find it for me. And again, I have the same ability here to change the axis. So if I look at my 8 peak beads on the fluorescein channel, I can see that quite nicely. And whether you use a dot plot or whether you use a histogram really depends on the question you're asking. I tend not to use histograms very often. If I'm doing DNA analysis, they're very useful. But for almost everything else, I will use some form of dot plot. So the other thing we have to think about right at the beginning is remembering that our flow cytometer has no intelligence of its own. They're not sentient, well, not yet anyway. So they're going to measure everything that is in your tube that's above the threshold. But of course, not everything in your tube is what you want. So immediately I look at this and I can see I've got some dots down here that are probably debris. I've got these doublets. So I'm going to want to reduce the data to the things that I'm interested in. And we do that by using what we call regions and gates. Now, they actually have a slightly different meaning, although they're pretty much used interchangeably now by everybody, including me, I suspect. But I suppose the real definition of a region is that it's a part of a plot, whether it's a bivariate plot or whether it's a histogram. But a gate is a combination of regions. So, what do I mean by that? Okay, I'll explain, hopefully. So what I'm interested in is this population here, my single beads. What I'm going to do is I'm going to set a region around those, and I have several options up here. So one of them is a rectangle. So I can just go and draw my rectangle. So FlowJo seems to think that everybody is an immunologist, so it defaults to lymphocytes. I'm just going to call that 1 for the sake of ease here today. So having set a rectangle around there, that tells me that about 91.7% of my events fall within that rectangle. One of the things that I think people, especially when they're new to the field, get a little bit anxious about is, how have I set that? The problem with light scatter here is that we don't have a control, do we? I've got no idea where that single bead ends and where it begins. So I'm making a bit of a subjective judgment. We like to think of flow cytometry as this fantastic objective technique, but there are some subjective parts to it, and setting a scatter gate is certainly one of those. But you could argue, why have you set it there and not maybe slightly smaller? And that hasn't really made a big difference to that. And if I were to give this data file, give control of my screen to Kathy or Rui, if he were here, and ask them to set a rectangular gate, they would set it in approximately the same place, but we may not get exactly the same number there. And that doesn't really matter. The point is, when you're doing your analysis, is particularly with this slightly subjective gating that we have here, is to make sure you're doing it logically and consistently. Now you might think, well, a rectangle, that's okay, but it doesn't really fit the shape of that very well. So let me get rid of that. And I could use an ellipse. So let me put an ellipse around that. Again, I'll call that 1. Pretty much the same percentage in there. Maybe you think that aesthetically looks a little bit better, but maybe we shouldn't be thinking of it in that way. I like rectangles and ellipses because they're very quick and easy to draw. I could spend a little bit of time drawing a polygon, a polygonal gate around that population, clicking at each vertex and joining it up at the end. But again, I get very similar percentage, and I've wasted 5 or 10 seconds valuable tea drinking time during that. I could do something even more free form than that. I could draw something, I don't know, like that, some fish shape around there. Again, the percentage is the same, and I'd probably get asked questions about why I'd drawn it like that. But you know, I do have that ability to have a free form drawing there. The other thing I can use is this thing that FlowJo calls an auto gate, which, as you can see, is like a bit of like the BD snap to gate, which some people like to think is maybe a little bit more objective, but you still have to make the decision about how big or small you make that. So there are multiple types of region I can set there. The important thing, if I come back over here, you can see that this region 1 that we set is slightly indented here. This is because it's a daughter, if you like, of the whole population. So this is the whole data file. And now I've created a region 1, that's a daughter of that. So this is what we call hierarchical gating. Now, if I were to double click in here, what I get is all of the data that's just within that region. FlowJo likes to give me an extra floating window, which sometimes hide in the background, which can be a little bit annoying. Now, if I were to look at, let's say, again, the 8 peaks here, and if supposing for some reason I'm just interested in these top 3 peaks, I can put a region around those, and I can call that top 3. And you can see that that, again, is indented. This shows me that I've made 2 regions that are now giving me a gate. And this hierarchical structure is probably the most common way that we're going to do our data analysis. However, most of our programs also allow us to do Boolean logic. So if I go back to this one, supposing, for example, don't know why we would want to do this, but we were interested in everything that fell outside that region there. FlowJo, I can click on it. I can go to workspace, or is it tools? Where is it? Tools, there we are. I can go to Boolean algebra, and I can make a not gate. So now I can look at everything that's outside the the region we've set there. Now, sometimes that is useful, sometimes it isn't, but it is useful to have that within the software. So if I go back up, I go back to here. The other thing I can do is I can set a region on my histogram. So I can do the same thing here. Rather than using that as a dot plot, I could set that top 3 peaks there as a histogram. Again, it's your choice whether you use a histogram or a dot plot to do that. Actually, while I'm on this plot, let me just remove that. Otherwise, we'll all get confused, don't we? And again, let's go for a histogram of my slightly cleaned up. So this is all of the data. So we've got that little doublet population. This is just our region 1 here. So one of the things that we talked about in the practical sessions was the way we can change the data scaling. So you can see here that we have some negative logs. So this is telling me that this is a bi-exponential or logical or whatever phrase you want to use for that. But this little T here allows me to change the scaling. So I could change this data to be linear. To be honest, this isn't much use to me other than to show that this looks like it's not very Gaussian. I can see the brightest peak, the second one, the third one, but I can't see the lower peaks because we're now on a linear scale. I could change this to a log scale, and I can see 1, 2, 3, 4, 5, 6 peaks. I can see the lowest, the negative peak, but everything's a little bit spread out. And a lot of flow cytometry is pattern recognition. So I want to make sure that I can see all of my data. So here I can go to transform this data. And rather than using a linear or a logarithmic, I can use any of the transformations that FlowJo has within here. Now, these are made to be the same as the transformations you would have in the software that you've acquired your data on. So if you've used a Miltenyi machine, for example, it has the Miltenyi algorithm in there. If you use the BD machine, which is what this was acquired on, it has the bi-exponential algorithm in there. And that's what we've just seen. So if I want to remove this or make this look a little bit better, I don't want all this white space here. What I'm going to do is just change this width bias, which is the amount of negativity. If you remember the bi-exponential scale, what that does is it's when the data is approaching 0, rather than using a log transformation, it uses a linear transformation. And I can reduce the size of that a little bit by changing that here. And that makes that data look a little bit nicer. It's not changing anything about that data. If I looked at the percentage of cells in each of these peaks, it would be the same whether it was on the transformed or the untransformed data. But now I can see my negatives looking a little bit nicer there. So the last thing I'll just briefly show you before we move on to a real biological sample is the output. So these windows, these floating windows we have, I mean, I can, if I go to Apple P here, I can print that, but that's not a very good printout, is it? So normally we're not going to do that. In order to get some sort of output, and I think Kathy will show this in a bit more detail later on, what we use in FlowJo is this thing called the layout editor, which looks a little bit like a set square on L up here. If I click on that, this is now a bit like a piece of paper. And if I drag my population over to here, I can see that. So I have now some ability to manipulate this a little bit. I can change the axis. So in the PC version, I think you can actually click on that and it will change the axis. In the Mac version, I have to double click and change that to, for example, the next one along. I can do things like change the color of that as well. And I have some ability to export that image or the multiple images that I have in multiple formats. So some of these are just image formats, as you can see, but we also have PDFs or scalable vector graphics, which are useful if you want to take this into something like freehand or Illustrator or something like that. I won't go into that too much. It's probably too much for today. But I wanted to introduce that layout editor because we will use that. I'll use that in a moment, and Kathy will use that a little bit later as well. So just checking. Okay, so yeah, that question, we will come to that question in a moment, I hope. So that hopefully has got us a little bit orientated, both within FlowJo and in the types of plots that we can use. So I'm going to remove that sample from here. I don't want that anymore. And what I want is my biological sample. And you'll notice this says viability. So what we're doing here is we're looking at the number of live and dead cells in my assay. So this is a real life example. It's from one of the practical courses that I've run over the past few years. So it's not perfect, and I'll show you why it's not perfect in a second. But let's think about how we would analyze this data. So one of the first things I want to do, and possibly in answer to the question that we had there, is we want to check the quality of our data. Before we spend possibly a long time analyzing this data file, I want to check that it's of reasonable quality. So one thing that we can do, if I double click on there, we have forward and side scatter. I'll come back to this in a second. But remember, in the header of our data file, in our FCS file, we have that time parameter. We know when we started acquiring the data. We know when we finished acquiring the data. So we can display this. I don't want to display it there. I want to display it on this axis. Doesn't matter which axis you actually display it on, to be honest, but you sort of get used to this. So I can display time against any parameter. Let me display it against, oops, not that one, forward scatter. So what I'm looking for here is fairly even acquisition throughout the time of my acquisition. So I'm not seeing any wobbling up and down here. I'm not seeing any data dropout. So I'm quite happy with the quality of that data, but I'm just visualizing that. If I go back to where we were before, looking at the inspector, I can also get this from within FlowJo, because FlowJo actually displays all of the parameters against time. What it's showing here is the median value for any of these particular parameters, and the dotted lines are 2 standard deviations away from that median. So as long as everything's within that, we're fairly happy. There are some R-based plugins that you can get from FlowJo, and we're not going to talk about those too much, but things like Flow Clean and Flow AI, which will do this for you automatically as well. And that depends, whether you use those, possibly depends on how complex or how automated you want your experiments to be. But it should be one of the first things that we do. I've got another example here that shows you something that's a little bit different. Oops, sorry, let me look. That actually that doesn't particularly matter. I want to look at this is from a data file from one of our users who said that they seem to have a bit of a problem with their acquisition. And here we've got a fluorescein. This looks like it might be a CFSE file. But if we plot this against time, what we see is that we've got a bit of an anomaly here in the middle of the data file. So I can go to that and I can say, well, actually, what that probably is, it looks like there were fewer events going through during that time. It doesn't look like it's changed the median fluorescence intensity of any of those peaks, but to be on the safe side, you might want to remove that. And the great advantage of having time as a parameter is that I can set a region on time here. So I can set that, and this is where the Boolean algebra comes in. I can set a NOT gate. So I can say I want to look at everything that's outside of that gate. So I've now removed the anomalous part of that data file. You might have been better doing that when you were running, but sometimes it only occurs to you to look at that when you come to the data analysis. But that should be really your first port of call. Get rid of that to make it a little bit easier. That should be your first port of call when you start your analysis, just to make sure that everything looks reasonable before you carry on. So let me go to forward and side scatter again. So I changed that to log. Let's go back to linear. So the question we were going to ask here is how many cells are live and how many cells are dead in my tube? And if we look at this scatter plot, many of you will be familiar with something like this. This is a Jurkat T cell line, nice round cells. And you might think, well, this is debris here. These look like it's probably mainly live cells. This is mainly dead cells. But we never want to just rely on light scatter to define viability. So we're always going to add a viability dye, and that can be a DNA binding dye, or it could be a protein binding dye. So one of the live dead amine reactive dyes that we can get. And which one you use depends on whether you're running your cells live or whether you're running your cells fixed.
Derek, I popped back up again just because we do have someone that asked a question about that time plot that you'd had. So they said that there was a line of cells at the very bottom of the plot around the edges. What are those and should we remove them? So if we go back to the time.
So here, there is a few things on the edge there. I mean, we can customize that axis, so we can change the number of seconds, for example, there. Maybe we need to go bigger. So all of the data gone too high. So nothing's off the end of the axis. So now we can see all of the data. The things that go off the end, yeah, we're going to come to that in a minute, because obviously we are going to try and remove cell doublets. Everything that's down here is going to have low side scatter as well. So if everything's okay, I wouldn't bother with a gate on this. If there's an anomaly, yes, I would gate that.
Thank you.
So let's go back to my forward and side scatter plot. Yeah, so we're going to add a viability dye. Sorry, I lost the thread there for a moment. We're adding a viability dye. Now, in this case, we've added a dye called propylium iodide. Now, those of you who know something about propylium iodide will know that it normally was optimally excited in this machine by a yellow laser, and it emits at around about 600 nanometers. So I would use this channel to look at it. But as I said, this is a real world example. It was done. What I do is I give the samples to a demonstrator and the students, and they provide me with some data. And if I change that to the relevant parameter, what we find is that a lot of our data is off scale. And remember, we talked about this in one of our other talks, webinars. One of the important things when you're setting the voltage on your machine is to make sure that nothing goes off the top of the scale. Because although I could get a percentage positive here, I wouldn't be able to have any idea of a level of fluorescence. But all is not lost, because we actually acquired every parameter on here. So we also use the blue laser to excite propylium iodide. And although we're not collecting it in the optimal filter, propylium iodide has a very broad emission spectrum. So actually we can see it at the 710 filter. And now, because we're collecting it a little bit away from its maximum, actually we can see that population on scale. So that's sometimes quite useful to keep other channels open, because you can rescue something. Now, if I click on this bit here, that duplicates the plot. And if I go back to side scatter, now what I want to do is explore my data, because I want to know, I've got lots of populations here, and I've got this one here, I've maybe got a little thing here, another one, another one. Where do they fall on the light scatter? And similarly, where do these clouds on the light scatter fall on this plot? So one thing I encourage people to do when they first start doing their data analysis is to explore their data. So I'm going to go back to the layout editor again here and just drag my, forward and side scatter plot. Sorry, just change that back to forward and side scatter and move that over there. Right. So with this plot here, what I want to do is see where these populations fall over here. So I'm going to set an ellipse, just for the sake of speed here, around this population. I'm going to call it population 1 again. So if I want to see where that population falls on this plot, what I can do is I can drag that population, just overlay it over here. So let me just change the colors, which might make it a little bit easier to see. So the red there are the events that are in that gate. And most of them in the scatter, as we might expect, fall in that big population there. But what about this population here that's brightly positive for PI? Hopefully you can see the dots there. Most of them fall in this cloud, and this is what we'd expect, because as a cell dies, its forward scatter tends to go down, its side scatter tends to go up. And because the cells are now permeable, this is why they're brightly positive for PI. What about this population here, sort of intermediate population? Again, most of those fall in that population, but you can see they've moved slightly to the right compared to here, because maybe these are cells that haven't quite become totally permeable, so they're maybe not scattering quite as much light as those very dead ones. Very dead. I'm not sure you could be very dead. What about this little population here? So now we've got some dots that appear in this cloud that, if by scatter, you might call that dead, but you've got some dots here that appear in this cloud that you might call live by light scatter. So now I probably wouldn't call those live if I were looking for a differentiation between live or dead cells, because they're slightly positive for propylium iodide. So death isn't an all or nothing phenomenon. Well, actually it is an all or nothing phenomenon, but permeability isn't. Cells don't suddenly become completely permeable to PI, which is why we have this difference, this gradation. What about these here? So these have low propylium iodide signal, but also low scatter, and they appear here in this sort of area. So as cells are dying, they're probably starting to break up, getting some debris, maybe with a little bit of DNA attached, possibly apoptotic bodies. And then if I go down to here, this stuff in the corner, that's my debris down there. So by exploring my data like that, I can get a better understanding of both the way these plots are displaying the data and also the biology behind it. Remove that for a moment. So the question we wanted to ask here is, how many cells are alive or how many cells are dead? So, I might be tempted just to say, right, these are my live cells. I've got about 60% live cells, right? That doesn't sound very good, actually. But it's not the right answer to the question, is it? Because remember, what we're getting in our flow cytometry is we're getting all the information from our tube. It's not necessarily what we're interested in. And what we're interested in, if I were doing this experiment on a hemocytometer, trypan blue, I'd be looking at cells, whether they were live or dead. These little bits of debris and maybe these sort of broken up bits of cells, I would be filtering out visually. So we also want to do that in our data analysis. So now, knowing a little bit about sort of the biology, I'm going to set a region to exclude everything that I don't think is a whole cell. So again, this is a little bit subjective, but I'm pretty sure that that's debris, and most of this is not a whole cell. So I'm just going to do something like that. We could argue the toss about where exactly I put those vertices, but biological meaningful name, cells, that's biologically meaningful. There you go. So those are my single cells, or are they? They're things that are whole cells. Remember, a flow cytometer can potentially measure two cells that are stuck together, and we want to look at that. So if I double click in here, what I'm next going to do is use a doublet discrimination gate. And again, this is something we covered in some of our previous webinars. I can look at the area and the height of any signal really, but forward scatter is a good place to start. And if I've got a single cell, the area and the height will largely be in proportion. So these are probably my things that aren't single cells. So again, I can set another region to include everything that falls along the diagonal. And I'm fairly generous with that normally. I try not to make that too close to there, because if in my next sample there was a little bit more variability, I might actually be losing cells that I'm interested in. Now, with this experiment, really, I just want to know how many cells are alive, how many cells are dead. I'm probably going to have a reasonable number there. Sometimes you're looking for a rare event, and in those situations, we might want to add another doublet gate. So I could double click in there, and one that I quite often use is forward scatter area against side scatter width. And maybe these dots here are doublets that I didn't manage to gate out with that first one. So if I'm really interested in rare events, I might put another region here, singles too. And now I can answer or try to answer the question that I was asked, which is how many cells were live. So we have 60% when we looked at the whole data file. So this time I'm actually going to make this a little bit bigger. Put one again to encompass those few things that might be blastic cells. So now I've got a more reasonable figure. I'm going from 60% to 70%. Still not brilliant, I have to say, but it's a little bit more sensible. So the very last thing I'm going to do before I hand over to Kathy is just mention something about saving your workspace here. So I can save my workspace as a workspace. Okay, and what that does is it puts the FlowJo workspace in the same folder as your data, which is great because FlowJo doesn't actually save the data in this workspace. What it does is it just saves a pointer to that data. So as long as you save your workspace in the same place, then that's fine. Next time you open up the workspace, that's brilliant. But if I were to send this workspace, if I were to email it to Kathy, for example, she could open it up and she could see this gating tree, but she wouldn't be able to see any of the data because it's pointing to my computer. So if you want to send the workspace and the data to anybody, what you have to do is save this as an archive or a .acs folder file. It's a lot bigger because it's containing the data, but that allows you to send that to other collaborators. Save that workspace.
And
Okay,
Derek, sorry to interrupt. We did have a question that came through actually about that second singlets gate. So Himanshu, yes. So Himanshu had asked, is it okay to use that combination of forward scatter and side scatter to gate the singlets since they're two different voltage pulses at the same time?
Yes, yeah, it works quite well. And I quite like using that because obviously I'm getting one signal from in front of the laser beam and one at 90 degrees. I mean, this works very well on the BD machines. I can't guarantee it works well on other machines as well. But yeah, combinations of area, height and width of any of those parameters works. And as I said, normally I would just use one, but if I'm looking for a rare event, I really want to be sure that that's a single cell. So we can add as many as you like to that.
Yeah. And in addition to fluorescence too, sometimes we can use.
Yeah, exactly. If I were doing DNA, I would use the fluorescence channel rather than scatter to do that. Okay, so I'm going to hand over to Kathy, maybe a few minutes later than I had anticipated.
Okay.
And I can take over in the Q&A.
All right, thank you so much, Derek. That was wonderful. So let me go ahead and start sharing my screen. And can everyone see my screen now? Derek, are we all good there?
We are. I can see
Okay.
that perfectly.
Perfect. Thank you. All right. So now that Derek has given us our introduction into analyzing some files in FlowJo, what we're going to do now is we're actually going to take the time to analyze a similar data set from one of our previous OpenFlow sessions. So as you can see, I already have the FCS files that are imported into FlowJo. And we actually have the file names that are coming up right here. And as you can see, what we're looking at here is cells that were stained with APC size 7. They were stained with an anti-human CD4 APC size 7. And as we can ascertain from the different file names, the voltages that these samples were acquired at were all different, right? So going back to a little bit of what Derek had discussed earlier, if we double click right here, what we can actually see is the voltages that the samples were acquired at. So if we're scrolling down, we see all of the fluorescent detectors were at 210 for this sample. If I go to the next, 240, to the next 270. So that coincides with the sample names right here, which is just what we want to see. Okay, so the purpose of this experiment for the analysis today is actually going to be how to determine using FlowJo what the optimal voltage was for this specific experiment and for this specific antibody fluor conjugate on this instrument. So what I'm going to do first is I'm going to double click on this first FCS file. And what you see here is that we have, just like Derek was showing, some debris in our forward and side scatter plot. And then also here, this population of cells, okay? These are actually lyophilized lymphocytes. So working in a flow core, or in my flow cores, we don't have any culture hoods. So these lyophilized cells wind up being really nice resource for us. I'm gonna use this polygon tool, and I'm gonna go ahead and create a gate that will include everything here and exclude the debris. So like Derek mentioned earlier, in case there's any scenario in which we might have cells that are a little bit bigger, blastic, activated cells, we want to make sure that we're being as inclusive as possible. So what I could do is I can close that gate off. And in this case, they actually are lymphocytes. So I'm going to leave the name and make it easier for myself. Okay. Now, if you wanted to tighten that up, if you did want to go ahead and only focus in on this population, that would be perfectly fine. And you could always adjust those gates after. It's based on your specific experiment and the application that you're looking at. So there's never going to be one answer that applies to everyone. Okay. Now what I can do is I can say, all right, I know that these are the same cells that I acquired across this voltage walk-up, and I want to make sure I apply this gate to all of my samples. So what I can do is I can click and drag and bring that up to all samples. And once I do that, this lymphocyte gate is now applied to each and every sample. Okay. I'm going to hold off on going into that for right now, but there are some pretty cool tricks for when you drag that up. But for right now, I'm going to double click on that first lymphocyte gate. And then what we're going to do is we're going to look at a scattered or singlet gate. Okay, so we'll look at forward scatter height versus area here, like Derek showed us before. And I'm going to choose the polygon again. And each click is a node. I might be a little bit more strict there. And that's just kind of how I'm approaching it, because I know for the most part, this should be a fairly uniform population, and these are the same samples, just run at different voltages. So I'm just going to be a little bit tighter there. And I'm going to leave that name of single cells and select OK. Now, this gating looks fine to me. And I'm going to go ahead and take this single cell gate and drag it under lymphocytes. So if I go ahead and put that gate for all samples, what winds up happening is it creates a separate gate. We want to be very careful, and this is something that often happens to people at the instrument. It happens to people in analysis. You can create gates, but if they're not in a parental gating strategy, the gates are independent of each other. So we want to be very careful that we place our gating in the appropriate way so that we're not looking at it separately. And we're making sure that from our debris exclusion, then we're looking at single cells. Okay. So I'm going to undo that change that I just made with this undo function. And I'm going to drag that single cell gate up to lymphocytes. Okay. Once I do that, that lymphocyte gate has now changed. Single cell gate has now been applied to every single lymphocyte gate for each sample. Then what I typically like to do from there is, once I have those two gates set up, if I select the shift key on my keyboard and I toggle through, what you'll notice is that both of these plots have just changed to the next sample. So right here on our scatter and right here on our singlet, if I click shift, we are now at 270. And I could very quickly go through and check each and every sample to make sure I'm happy with that gating. That's something I like doing as I'm going along, because it's a quick verification that all of my gating is appropriate and everything looks okay. Now, if you notice, if I say, okay, you know what, this sample, maybe something happened and I need to adjust this gate. It's a poor adjustment, but just for purposes of showing everyone how this works, you can make any changes to individual samples as necessary, and the gate will still be there. It's adjusted, but it's no longer bold in the workspace. That's perfectly fine. It just shows that it's not the same as the original parent gate that was created there in the hierarchy, okay? Now, if I say, all right, that was a mistake, I didn't want that, all I have to do is select delete, and yes, and it snaps back to where it originally was, okay? So that's perfectly fine to do. And then from there, what I want to do is I want to look at my analysis of, or I want to analyze rather, how that voltage walk-up worked for us. So I'm going to open up our single cells, and I'm just going to do the one single cell gate for this analysis here. And I'm going to start off at 210 voltages. So here, I'm going to change forward scatter area to APC size 7. And what you see here is there's a lot of white space here. Derek reviewed this a little bit earlier, so I'm just going to go ahead and reiterate how we adjust for that. Hit the transform button and customize axis. And when we do that, we can adjust the width basis to really show the distribution around 0 more appropriately for our experiment. When I click apply, we can then see that the population resolves much better, okay? So here, all I really see is one population. I see this one population of cells that are coming up, and I'm not really seeing a negative and a positive. Remember, these are lymphocytes that I've stained for a CD4 antibody, so we should expect to see a nice robust population, okay? Keep in mind, this setting that we're at for this detector is very low. So 210, as far as voltage goes, is pretty low. So I can't gate right now on a positive and negative. But once I click on this next arrow, what you can now see, I'm going to make this a little bit bigger, is we can see a negative and a positive population. Okay? Now, to Derek's point from earlier, if we go ahead and change this to histogram, it becomes very difficult for us to decide what's positive and what's negative because of the distribution and because it's a univariate plot. Once we change this to scatter or potentially change it to another detector that we have open like FITSI, and then change that with basis there as well, we could start to see if we could pull out the population better, okay? So here what you see is we have really nice, well-resolved populations, and we can gate on them. So what I'm gonna do is I'm gonna go ahead and just choose a rectangular gate, and this is gonna be our negative. And then I'm gonna choose one more for our positive. Okay, so now that we've done that, what I can then do is I can highlight both of these gates right here and drag it under the single cell population. What's going to happen as we go and we increase our voltage and we go to our next sample, you can see it's resolving a bit better. What I see is I see 57% of cells that are negative and 39% that are positive, okay? If I go back to this 240 sample, it's not looking exactly the same as far as percentages go, and that's very likely because we're at such a low voltage, we're not yet pulling out the best resolution as we can for these samples, okay? For these populations stained with the APC Cy7. So I'm gonna go ahead and go to next again, and I'm just gonna repeat this process. And what we should see once the populations become discernible is that these numbers should not be changing drastically, right? So once we're able to resolve the positive and the negative, these population percentages should be somewhat similar as we're going through, because remember, it's the same exact sample. Okay? So this is going to be a little bit boring for everybody who's watching, but remember, you can ask any questions if you have it. And I'm just going to go ahead and I'm going to adjust these gates. And the other important thing, and what might be a question as people are watching me do this, they might say, what is this intermediate and why are you not gating on it? So what we have to remember is that staining for CD4, you can have moderate CD4 expression when you're looking at PBMCs for monocytes, because they can have that expression of CD4. For this purposes, we're just looking at the lymphocytes for the purposes of this experiment. So that's what I'm focusing on here. I'm going to keep going along and adjust these gates. Each one and pull out those populations of interest. Okay, now we could also explore the use of different gates, right? So we could explore the use of polygon gates, elliptical gates, or freeform, right? So we have those different options, but we're perfectly fine using this for this analysis today. The other thing I want to point out is that if I come here and I go around this population, there is going to be a distribution that of cells that we're not catching. So I want to bring it against the axis. So by going ahead and capturing that population that was to the left, went from 39% to 44%. What I could also do is I could select the transform button and customize the axis and adjust that width basis. And that is going to change, right, as we're going ahead and doing our voltage walk, because we're going to see a little bit of a different spread of that negative. Okay, so that's okay to do. At first, I had to adjust it to be able to appropriately set the gates for our lower voltage samples. But as we're going along, I have the two options, and my two options are to either adjust that width basis or alternatively, just make sure I'm against the axis to collect all of those negatives.
Hi, Kathy.
Yeah.
I'll just stop while doing that. There's a question here about, could you just explain again the purpose of the voltration and antibody titration?
Sure. So those are actually two different things, right? So what we want to make sure that we think about is at the cytometer, we have the capability to set fluorescent detector voltages. So when we set fluorescent detector voltages, if it's set too low, what's going to wind up happening, as we saw from the 210 volt sample, is that the amplification that's applied to, in this case, a photomultiplier tube, is so low that we can't actually pull out the positive from the negative. So what we want to do is we want to do a voltage walk so that we can determine what the appropriate settings are for the instrument, where we get the ideal resolution and the highest stain index to pull out the positive from the negative. So that's what this experiment is, and we're analyzing data from that. Antibody titration is also very important, and that's when we're adjusting the concentration of our antibody to make sure that you're actually at saturating capacity and you're not seeing an increase of the negative from non-specific binding. So that's when you titrate from maybe 10 micrograms per mil and do 8 serial dilutions down and determine the optimal staining concentration.
So I suppose one question that I think we might have had before as well is that it's a bit of a chicken and egg situation, isn't it?
Yes.
Because you need to have this titrated really before you do the voltration. So.
Exactly. And what people can kind of consider doing as well is, sometimes if you're a little bit higher as far as the voltage goes, as long as your brightest sample is on scale, you can then go ahead and do an antibody titration, find your optimal concentration for the antibody itself, and then once that's optimized, go ahead and do the voltage walk up with the appropriate settings for the, and make sure that you find the appropriate settings for the experiment after you calculated the optimal concentration for the antibody. But it is a chicken and egg, absolutely.
Thanks.
Thank you, Derek. Okay, so from there, we don't have any more samples to go through. So you guys were very patient in watching me go through and adjust all of those gates. We stopped the acquisition at this point because, as you can see, the positive sample is just about on scale still. If we had bumped up the voltage anymore and went to about 690 or 700, that signal is going to be off scale, and then it's no longer reliable for statistical analysis, okay? So this first tick after 10 to the 5th is 200,000. Typically what winds up happening on the photomultiplier tubes on these instruments is that they're linear to about that range or higher, but 200,000 is a good solid number, and that's where that first tick after 10 to the 5th is. So that's where I typically tell people to make sure your signal is right about there or below. And similar to what Derek mentioned before about the stats and everything like that, you also can wind up with multiple intensities of your positive. So as we see here, we have this moderate and this high CD4. If all the positives were off scale, we would just be able to get a number of percentage, right? Like a percentage number that says this percentage is positive. I would have no idea that there was a moderate and high expressing if all of that data was off scale. So that's very important. Okay, so now that I have that analysis done, what I can do then is I can close this plot right there, and I can close these. And what I want to do is I want to go ahead and touch on this layout editor. From the layout editor, what we can actually go ahead and do, scroll up to the top, and I can drag this FCS file in. and it's going to automatically show us that first gate. From there, I can drag in the lymphocytes gate. And once I do that, it's going to show me what was gated on lymphocytes and show me that singlets plot that I created, okay, and gate that I created. If I drag in the single cell population, it's then going to show me the negative and positive gating I made with APC, Cy7, and FITC. Little things that I wind up doing during my analysis is I'll go ahead and delete all of this right here at the bottom because I don't want it. And you could go ahead and, for the purposes of a cleaner analysis, insert a text box. And when you do that, if you right click, you can insert any keywords that are specific to the file. So this is the first sample, which is 210. And if I click on that, some of those keywords that were mentioned before now come up on this list. And if I go down to the bottom, I can select tube name. And now I know that that's my 210. And I'm very well aware that it's a very small font size and we have people on various devices that might be hard to see. Okay, so I made that a bit bigger. Then what I could do is I can go ahead, if I'd like, I can adjust the page view right here. And we can go ahead and create a batch report so I can get an idea of what all of that data looks like for all of my samples. So if I want to go ahead and look at all of this in one, for all of my samples, what I can do is I can go and select one column down and create a batch report in this layout editor. And when I do that, what's going to wind up happening is we're going to see every single sample. And you could use that to double check the gating and make sure everything is appropriate. Okay, so as you can see here, everything looks good. Happy with that. And then you can also go ahead and print it to a PDF. You can go ahead and export it to a PowerPoint. When you go to do that, there are multiple options here for you in the software. Okay. So that's one way that we can use the layout editor. And I'm going to minimize this right here. Yeah. Unless Derek has a question.
Yeah, I have a question from an attendee before we get on too much to the maths and things here. Is it okay to do vultration with antibody capture beads, do you think? Especially if you don't want to buy CD4s or CD8s every color you want to look at.
Yeah, so that's something that people often do. Just be mindful that you want to be a little bit, you want to be considerate about the autofluorescence of your cells, and you might have cells with varying levels of autofluorescence or trickier cell types. So it might give you a good starting point, but it would likely be best on your cells of interest and your experiment and the panel that you're working with, just to do a quick optimization. Not to say it has to start at 210 and go up to 660. You can probably start at a higher voltage. But just if it were up to me and if it were my experiment, I would probably try and work on my specific cells to make sure I have the best separation. Yeah,
I think if you're doing lymphocytes, it's probably reasonable to do it like that. If you're getting anything different, no, it possibly isn't.
Yeah, exactly. And especially for cells that, that's my focus, cells that have varying levels of autofluorescence where it might be a little bit tricky, to definitely try and optimize with the cells. Okay. Any more questions did any more questions come through?
No, that's fine for the moment, Kathy. Okay. I'll interrupt you again later.
Awesome, thank you. Okay, so from there, I'm just going to minimize this layout editor and we can come back later if we need. And what I want to focus on at this point is the statistical analysis. So FlowJo gives us the capability to carry out statistical analysis. And going back to an earlier question about analyzing your samples in DIVA, it's not as straightforward. It's not as easy to do a lot of things in the acquisition software. And that's for many acquisition softwares, not just one. And we can utilize these analysis softwares specifically to help us do an easier analysis and statistical analysis. So what I can do is I can go ahead and go to the workspace tab right here. And if I click on statistics, you can see there's the option to add statistic. And if I click on that, it gives me a whole bunch of options here. Or if I click, it gives us some of the most common ones. So if I want to add a median for example, I can choose from this list right here. And if I were to do that right now and say I want it for this APC size 7 detector, I immediately see that it went under my single cells. That's not what I want to do, okay? What I want to do is I want to do a statistical analysis of my positive and negative populations in order to calculate stain index. And what the stain index is, for anyone who's not familiar, it's the MFI of the positive population minus the negative MFI over 2 robust standard deviations of the negative. So I'm going to create those statistics for those populations and show us how we can do an analysis here of that. So if I go to statistics and I go to median, I can go ahead and choose APC size 7 for our negative, and I can do the same exact thing for our positive. So what we're seeing is we have an MFI of our positive and negative, and I just want to note that it will give us stats for anything that is within that gate. So even though I really did not see much in that positive gate here for this sample that was at the lowest setting, it still does give me a statistical readout because there were 49 cells in that gate. And just be cognizant that if you do want to remove any samples from your statistical analysis, you might want to remove it from the group or from the workspace, okay? But I'll leave this in for now. And then what I'm also going to do is I'm going to add in that robust standard deviation of the negative population. So I go to statistics and I can add a statistic here. And what I want to look at is the RSD of the negative population. So that's right here. It's already selected on negative. And I'm going to do APC size 7. Add that, and then I can close. Now you have the option of dragging this up, but you don't need to do that because all we need to do to create our a table of statistics is go straight to the table editor. So the table editor is where I'm clicking right here. And once I do that, I can specify to FlowJo what statistical analysis I want to look at. So I can say, I want to look at my MFI of the positive, the MFI of the negative, and the RSD of the negative, and drag that in. And now all of those stats are here in this table editor, and I can create a table. So for example, if I want to create a table to display to a file, which can be text, CSV, Excel, so on and so forth, to the layout, you have those options to pick from. I'm going to choose display and create a table. So it tells us here on this table that we've displayed what our tube name was and what the stats are for each and every sample. Now, those are great stats, the MFI, the positive and the negative, and the RSD of the negative. But what I don't have is the stain index, okay? So I actually wanted to use those statistics to calculate what the best voltage was for this experiment. So I'm going to close that out and show everyone here how we can create a formula. So if I go into edit, what we can actually do is we can add a column. And in this column information, we can go to the formula tab. And I can say, all right, for this column that I just added, that's going to be my stain index. And here, I can use the statistics that I just imported into the table editor to create a formula. So I'm going to do 2 parentheses to start, and we can insert the reference. And when we do that, we can pick from the statistics that we've imported into the table editor. And I want to look at the MFI of the positive minus the MFI of the negative. Then I can close that off. And then what I'm going to do is I'm going to have that divided by, sometimes I add too many parentheses, but it works. So I'm not going to critique myself on that. 2 times the robust standard deviation. Okay? And I'm going to close that off. You can see if you ever have it incorrectly where you haven't correctly bracketed any of your information, there's going to be an error in the algebraic expression. So we just close that off, make sure all of that looks okay. And then I can select OK. and we have that stain index formula there. The other nice thing we can do is we can add keywords. So if you want to say, I want that tube name in there, all I have to do is start typing in some credentials that I know or the starting name of that keyword. And there it is. We have the tube name that'll come in for the table editor, or for the table that we create rather. I probably don't need that, but I'll leave it in, and I can always drag it up to the top and change the order if I want. And then from there, what I'm going to do is I'm going to create a table. And here, what we see is we have our tube name, we have our MFI, positive, negative, RSD of the negative, and here you can see the stain index that was calculated. I can also go ahead, like I said earlier, and create that to a file. And I could do Excel, CSV, and tell it exactly where I want it. So I went a little fast there. So here in the destination, if I click on that icon, I can put it right on the desktop and say I can name it OpenFlow is awesome. I hope it is at least. We click save, and I can create that table there as well. And what should happen is it should go somewhere on my desktop. So right here, I tried to clean up my desktop, not as good as Derek did, but my very best. And if I open that up, it opens up the table that we created in FlowJo. And then you can do fun things like creating tables. So if we look here, this is an example of a table that we can create in Excel very easily based off of those statistics that we exported. So this is that same set of data that I previously analyzed, and you can see here how we can generate the stain index versus voltage and see that optimal point where as we're increasing the voltage, the stain index increases, which means we're getting a more optimal resolution. And at a certain point, which here is about 510, we hit the maximum for stain index, and then it tends to plateau. And that's something that's often brought up where people think just increase the voltage, increase the voltage, increase. It's at a certain point not necessary because we've already reached the best stain index we can, and then you're just increasing for no reason because we're not getting a better resolution, okay? So there are a lot of caveats to that, and we can always have good discussions on, when it's appropriate to increase or decrease. But for these purposes and for this experiment, you can see we got to that optimal setting, and then it just plateaued, okay? Now, what I also do want to quickly show is that I did all of that manually, but there is something in FlowJo called a plugin. And for anyone who is somewhat familiar with plugins, this might be useful to know that there is a stain index plugin. So I'm not going to go through the whole rigmarole today, but if we go ahead and go to Workspace and select plugins, there actually is a stain index plugin. We want to make sure that the workspace is saved. And if I select yes, go ahead and save that here. And what I can do is I can say the stain index parameter is APC size 7. Our positive gate, we can either have it auto find the gate, or what we can do is we can say our positive is what we already dictated as positive and our negative is what we dictated as negative, and select OK. And I said I wasn't going to do it, but I just did it. And then what it does is automatically comes up with this formula that's going to be exactly the same formula that I created, just with less parentheses, like I mentioned, and it'll give us the stain index, okay? And then we can apply that to all of our samples. Another thing that's just a nice visual that I'd like to wrap up with, because we're about to run out of time, is how to visualize that voltage walk in a nice way. So what I like doing is I like taking a look at my single cell data from each file and concatenating it into one so I can get a nice visual of what that voltage walk looks like. So I'm just going to show you some shortcuts. So on my PC, if I do Control-Shift-E, it goes ahead and it captures every node of the same, or it captures the similar node from each and every sample. So the same single cell gate. And I could right click and I can concatenate or export the population. And that would have been Control E. And then from the populations, I can choose to concatenate them and say where I want it to go. So if I want it under desktop and under the file where the folders are, I can say that's fine. I could rename it and say I want that to be my APC size 7 concatenation. Okay. And then from there, I can concatenate the file into the existing workspace. And I hit close. And I like doing this just because it's a nice, pretty way to see at the very bottom. If we look up and look at our single cells, on the x-axis, I could do sample ID. And then here on the Y, we can look at APC size 7. And then what we see is this really beautiful representation. As we look up our voltage, you see that we have the positive coming up, but then we also see that the negative is starting to spread and shift up as well. And we can go back and look at that analysis that we had based on the statistics, and we can see the point where we have the best possible stain index and compare that to what we see on this plot. So it's a nice little tool in FlowJo that we could use. And with that, I think I've gone through pretty quickly and exhausted my time, but that is a very brief introduction to doing a voltage walkup analysis in FlowJo.
Okay, so we have another question, Kathy, that we can probably both answer. But so do you do these optimizations yourself for your users in the core? And if you don't, how compliant are people before they do their experiment? I'm sure we can both answer that one.
That is a wonderful question. So I will say that it is a bit labor intensive because you have to do it depending on how many instruments that you have and depending on how many parameters there are. That's a lot of staining that you have to do. There are alternative methods where you can use the peak 2 method where you have unstained cells and a hard dyed bead that you can go ahead and do a voltage walk up with, which is something I'm going to be testing out. But previous institute, we went ahead and did this on all of our instruments and have it available. So at Memorial Sloan Kettering, where Rui's at, we have all that data there. I plan on doing it here at the Whitehead in the next couple of weeks, since they just came on board here. Advise everyone to do it for their experiments, but also understand that it can be very difficult to do. So I mentioned to people that, you can do this and it's absolutely recommended. But if you can pull out populations and it's very clear, it might not be necessary.
Yeah, that's exactly it, really, isn't it? I mean, the whole point of getting to the right sensitivity is it gives you the best resolution of anything that's weakly positive. If you've got a panel where everything's screamingly positive, you can probably get away. The tolerance is a little bit better. But certainly if you were doing a large panel where some of your expression is not very high and you were doing it over multiple months or years, I would spend the time optimizing this before I started that experiment.
Yeah, absolutely. And also understand that every instrument's different as well. So different instruments have different lasers, laser powers, optical configurations, and want to, really stress that just because you do it on one instrument, that does not mean you'd run it at the same settings for the PMTs or avalanche photodiodes, whatever your instrument is and whatever the detectors are, optimize it for the instrument.
And cells are different as well, of course.
Yes, yes.
Okay, so,
all right.
Oh, hang on. All right, so there's one more question. I'll see at the bottom there. Sorry, it says, I've lost it again. Right, how do you ensure the optimal voltage obtained from filtration doesn't complicate your compensation in later panels? That is a good question.
Yes, we're ending off with a very complex question, I would say.
Yes.
Yeah, so what you want to do is you want to optimize your voltage to get the best separation, the best stain index for the individual detectors. And then if you go ahead and you run your full panel and you see that there are issues with spreading or any issues there, you want to consider optimizing your panel likely, because what winds up happening is when we see spreading, which is, not compensation, it's an issue with the the photon counting that we have at the instruments, typically there you'll wind up seeing issues. Compensation and high compensation values are okay. You will see that there are going to be educational resources that show compensation over 100 where you get nice populations that are pulling out. Not everyone might agree with me on that, right? So
I do.
Yeah, I know there's some people that have different opinions, and some people try optimizing voltages to bring down compensation, but that's not the approach to take. And we have some educational resources on that.
Yeah, the most important thing is making sure you're getting the best resolution of your populations, and the number, the compensation value is pretty irrelevant after that. In fact, sometimes you can have high compensation values and no data spread. And data spread, again, is more important than compensation as well. So yes, you have to consider all these things, but the compensation value, I think, is probably low down the list. You really want to make sure that you can separate those populations.
Exactly.
Okay, so can I just grab the screen back?
Sure. Let me go ahead and I'll stop my sharing, and thank you everyone for the great questions.
So we just got just a couple of wrap-up slides, which you can't see because I've lost PowerPoint. There it is. Sorry, so this is the disadvantage of having a small screen and moving things around to see what I'm doing. Okay, so obviously, if there are any outstanding questions, get them in the next 30 seconds or so. And we hope you found that useful. And as usual, we will send around a questionnaire, or we should get sent around a questionnaire at the end of this. It's very short. It's great if you could spend a couple of minutes to fill that in, because it does help us with thinking about what to do next, not to do in our next series of webinars. This has been recorded, so it will be made available on the OpenFlow Cytometry YouTube site, probably towards the middle of next week. And if you enjoyed it and you want to come back again, please do, because our next date is already set. That's June the 25th, which is 5 weeks today, same time. So 13 or 1 o'clock, 1 p.m. EST or 6 o'clock BST. And we're going to look again at flow cytometric data analysis. We will be using a different program. We'll use FCS Express in that one, and we'll look at a couple of different types of analysis. So registration is already open. It's a very complicated URL there, but if you just go to www.cric.ac.uk/what's-on, you can register through that. And of course, registration is always free and open to everybody. And if you do want to contact us, you can reach out to any of us individually. Those are our email addresses, or you can use the OpenFlow cytometry@gmail.com address, which will come to all of us. And then you'll probably get 3 answers, which are hopefully the same. But depending on which time zone you're in, you might get an answer a little bit more quickly. And of course, you can follow us all on Twitter. And since our last one, Kathy is now established on Twitter, WIBRFlow. So you can get in contact with us in many different ways. And surprisingly, we've only overrun by about 5 minutes, which is pretty good for us. So thank you all for coming. Enjoy your weekend. And if you're not vaccinated yet, hopefully that'll be coming up for you guys soon. So thanks very much for coming. We'll see you all in a few weeks. Bye, everyone.
Thanks, everyone. Bye-bye.